This is the Professor, CBMD PhD. It's Saturday, August 8th, 2026. It is 5:14:30 p.m. Eastern Standard Time. And today's research, immunotherapy. So let's hop to it here. A protein older than blood circulation can transform cancer immunotherapy. An ancient immune molecule may help turn stubborn, treatment-resistant tumors into targets for cancer immunotherapy. Date August 8, 2026. Source: the Gojo University. A protein that evolved long before the human circulatory system may help unlock stronger cancer treatments. When C3 is produced inside tumors, it blocks immune-suppressing cells and gives immunotherapy a better chance to work. Researchers were able to recreate this effect in resistant tumors, significantly improving survival in mice. C3 is an evolutionary ancient protein found even in simple animals such as sponges and jellyfish. Most C3 is made in the liver and released into the bloodstream, where it helps the immune system defend the body against infections. Scientists know much less about what C3 does when it is produced directly inside tissues and organs. Cancer tumors are surrounded by normal cells called fibroblasts. Until now, the role of complement C3 produced by these cancer-associated fibroblasts within tumor tissue was not known. The researchers found that C3 made within tumor tissue prevents immunosuppressive myeloid cells from entering the tumor microenvironment. These cells can weaken the body's ability to attack cancer. By keeping them out, locally produced C3 gives the immune system a better opportunity to recognize and fight tumor cells. The results point to C3 as an important regulator of cancer immunotherapy, a form of treatment that helps the immune system identify and attack cancer cells. To determine whether C3 in the bloodstream also affected immunotherapy, the team conducted experiments in mice that allowed them to distinguish between C3 from different sources. When the amount of liver-produced C3 was reduced by 90%, an immunotherapy drug anti-PD-1 antibody remained just as effective as it was in mice with normal C3 levels. The outcome changed when researchers stopped fibroblasts inside the tumor from producing C3. Under those conditions, the same treatment became less effective, even though the amount of circulating C3 fell by only a small amount, a 9% decrease. What determined the efficacy of the immunotherapy treatment was not the C3 in the blood, but the local C3 produced at the tumor site. When this C3 breaks down, it forms a fragment called IC3b that stops harmful myeloid cells from entering the tumor. As a result, immunotherapy is more likely to work. The team then investigated whether they could reproduce the effects of C3 in cancers that normally resist immunotherapy. They tested a drug designed to imitate the way C3 blocks myeloid cells from entering tumors. This approach allowed immunotherapy to work against tumors that had previously resisted treatment. It also significantly extended survival in mice. The findings could eventually help doctors identify which patients are most likely to benefit from immunotherapy. It may also lead to new treatment options for cancers that do not respond at first. Researchers also examined tumor samples from patients with lung cancer. Those with higher levels of C3 in the tissue surrounding cancer cells experienced better treatment results and longer survival. About half of the patients with high local C3 levels responded to treatment. None of the patients with lower levels responded. As in the mouse study, C3 levels in the bloodstream were not linked to treatment success. The researchers now plan to test ways of increasing C3 levels inside tumors and to determine the most effective timing for treatment. They also believe that learning more about the local activity of C3 could improve understanding of other biological processes, including wound healing and the regulation of inflammation. And this videography was created for educational purposes only. My code name is Will. I'm Professor Christopher Brunet, leader of research scientist, ufologist, psychic, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. My passion is world peace. Have a great day. This is the Professor, CBMD,